food, drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. Visit mindandmatter.substack.com to find all of my content. Professor Michael Snyder, thank you very much for joining me on a Sunday morning.
Great to be here.
Do you want to start off by just telling everyone a little bit about who you are and what your lab studies in general?
Sure. So I'm a professor of genetics, although we study use big data to study lots of biological problems, mostly health. We're actually very interested in trying to profile people's health, catch disease before you become symptomatic, and really try and change the way medicine is done today.
And one of the reasons you're interesting is you study things at multiple levels. You do work in humans, you do work in animals. You get down to the nitty gritty in terms of mechanisms and molecules, and then you do other types of research as well. So you've got a wide swath of research that you've done.
We do, but we're mostly humans these days. I think profiling, people, and then to be honest, we just use mice when we go try and further get into the mechanisms.
Yeah. But as someone with that kind of background who can take that kind of broad view and has that range of experience at a high level, when we're thinking about human health, when we want to think about human health and think about, how does this food affect a human? How does this disease work in a human? But we're thinking across all of the literature, and we're trying to take an integrated view of how do we think about the mechanistic stuff and the animal stuff and the clinical stuff, thinking about the pros and the cons and the limitations and the strengths of each. What is your general approach as a scientist to sort of considering all of the literature holistically and sort of balancing the pros and the cons of the different types of research when you're thinking about what's true for human biology?
Yeah, I guess this is my bias by the kind of approach we use, which is we tend to, as I say, profile people pretty deeply. And by that, I mean, we'll sequence their genome, we'll measure as many molecules as possible out of blood, urine, their microbiome, and such. So we're deep, deep measurement people. So I guess I tend to go into biological problems like humans and human nutrition from a we don't know anything standpoint, which is to say that we try to discover what's there, let the data tell us what's going on. And that has turned out to be reasonably successful. So, for example, and we may get into this, we've run a fiber study where we have people eat different fibers and look at their response and profile fairly deeply. Now, that's an easy one because if you read the literature on fiber, it's very conflicting. And we all know, for example, fiber is good for us. But there are many different kinds of fibers. There's short chain, long chain, hydrophobic, hydrophilic, positive, negative charge. It's all over the map. It's like saying all animals are the same.
Right.
So we think that, so what we do is we'll go in and we'll test two different fibers. In this case, something called arabinoxylan, which is in Physium husk, Metamucil, things like that. Another one, inulin. These are very popular fibers, and we'll see exactly what's going on with each of these when people take them as supplements. And by looking at their response, we then try to figure out the mechanisms. And I guess this is what both takes us broad and takes us deep in the sense that when we see responses, for example, arabinoxylan actually does reduce your cholesterol. Now, you'll say, oh, the literature has shown that, but I can show you other studies where the people say fiber doesn't, it was conflicting when we went into this. So anyway, this is how we approach things. And then if we want to study it further, which we do, we'll start testing some of our ideas when we make these discoveries. We'll test them on mice and see if we can see the same results. But in the area of nutrition, it's very, very tricky because what goes on in a mouse may not happen in a human. It's really a problem because mouse metabolism is obviously very different from humans. Some things, basic mechanisms do cross all organisms, but a lot of the details will differ.
On the subject of human variation to diet, you did a study recently that looked at individual variation in the glycemic response to carbohydrates, and you dug into that in many different ways. But the basic idea here is not everyone is necessarily going to respond the same way to a given type of food that they ingest. So I want to start with what the glycemic response is and then dig into what you guys found there. So let's just start with the basic definition. What is the glycemic response and what are we measuring when we look at that?
Yeah, sure. So when you eat anything with carbs, there's a good chance your sugar will go up. And if you have good glucose control, your insulin gets made from your pancreas. I'm sure everybody knows this, but your insulin gets made. It stimulates your various cells to take up the glucose, and you maintain a nice, good glucose control. And when we're young, we all have very good glucose control. At least most of us do, not all. But glucose can go out of control. And the way diabetes is classified these days, it's usually two forms. Type 1, where you actually have a problem in making insulin in the first place, because usually from autoimmune disease, and type 2, which turns out to be very, very heterogeneous, and people don't appreciate that. We think this is a huge, huge deal because some people will be muscle insulin resistant, some people will be hepatic insulin resistant, adipose insulin resistant. Some people are beta cell defects, meaning they don't release insulin from their pancreas. Other people have defects in incretins. These are these GLPs, which are super hot these days. And some people have defects in those. And so what we've been doing is going in and subtyping diabetes and actually, or in this case, prediabetes. We're actually trying to keep people from getting diabetes. And so we try to profile people while they're so-called normal or pre-diabetic. And what we discovered early on is that different people will spike to different foods. So meaning some people spike to pasta, some to potatoes, some to white bread, others to brown bread. It's all over the map. Very, very personal. And Aaron Siegel has shown that about 21% of this is due to your microbiome, but most of it's not explained. We think it's actually due in part to the type of diabetes you have, or glucose dysregulation, I should say, because a lot of these folks aren't diabetic yet. But I maybe backing up a little bit, just a problem.
But I mean, already what you're getting to here is it's not just type 1 and type 2 diabetes. Even within type 2 diabetes, there's lots of variation and there's probably distinct subtypes. And the question is, what are they and why? Do people's ability to regulate glucose, does it break in different idiosyncratic ways? Or are people just basically born with different types of biology that will then enable type 2 diabetes to manifest in different tissues or in different ways? And the question is, one of the questions would be, even before someone's diabetic, they're pre-diabetic or even healthy, are they responding to glucose? Are some glucose metabolism working differently in different people for whatever reason?
Yeah, there's a lot to unpack in what you just brought up. But the answer is yes. Type 2 diabetes, of which 90% of people are, is very, very heterogeneous with these different subtypes. This muscle insulin resistant, meaning your muscle cells don't respond to glucose. Hepatic insulin resistant, same sort of thing. Beta cell defect, as I say, you make insulin, but you don't release from the pancreas. I have a beta cell defect. Now, most people look at me and say, "There's no way you're type 2 diabetic," but I am, actually. And then to the issue, what's the cause? Well, it's not always clear. In the case of type 1, it's an autoimmune disease. In my case, it's very fascinating. I became diabetic. My genetics predicted I'm at high risk, but I didn't become diabetic till after a nasty viral infection, a respiratory syncytial virus, which is a pretty common virus, actually. And I got fairly sick, and then bam, my glucose went out of control. I got it back partially under control, but then it came back after another viral infection. So in my case, and by the way, that's now showing up in COVID. 2 to 4% of people get COVID wind up as diabetic. So this is hypothesis, but one idea is that your pancreas is very sensitive to stresses, and things like viral infection. They can be pretty severe stresses when you get quite ill. And I suspect that your pancreas, which is a strong overproducer of insulin, it really takes a lot of energy to these cells. That's their main job, crank out lots of insulin. I think they're very sensitive and can go off. And so that's what exactly that means mechanistically is not so clear, by the way. In fact, all this is a little bit conjecture. But the bottom line out of all this is that it's very clear, we are very heterogeneous. And we think that's important because it affects your glucose spiking. And that's this paper you were bringing up. It turns out that what we did was we put a bunch of people under the exact same amount of carbohydrates, but the carbohydrates were different form.
Yeah. And are these pre-diabetic people, healthy people? What's the population?
Yeah, they were normally normal or pre-diabetic. One diabetic snuck in there who was in our study, but in general, they're pre-diabetic. And most people, by the way, are pre-diabetic, don't know it. 80 to 90% don't know it. And yet they will become diabetic within 5 years.
They're on the path to get there, but nothing has gone so haywire that they've had to go into the doctor yet.
Correct. And they don't even know it. They don't realize it. And again, it's very common. So I'm a big believer people should be getting tested for this stuff because they're running around with some glucose dysregulation. And then bottom line is, that based on what we did was we had people eat these different carbs. We discovered that different people will spike to different carbs. Some people are potato spikers. Some people are bread spikers. And most people, by the way, are rice spikers. People don't realize that white rice is probably more glycemic for most people than ice cream.
Wow.
Really, it's a strong spiker. Yeah. It's really quite fascinating because Asians turn out to be even more spiky than non-Asians.
Yeah. Well, let's come back to that. But the idea here is, right, you just put a glucose monitor, like a blood testing thing that some people you see wearing around might have on. You guys put this on your patients, on your test subjects here, and you're measuring how big the glucose response is, how long it lasts to different foods. So you got a bunch of people, you give them rice, you give them potatoes, you give them bread, this, that, and the other, fruit, and you're measuring how high does that glucose spike, how long does the spike last, how long before it goes back to baseline, those types of things.
You got it. So that's exactly right. So first of all, I recommend everyone try these. And you can now get them over the counter in a drugstore. So they'll measure your glucose every 5 minutes. And you'll be amazed. You'll never eat the same again. And I mean that in a good way, because what'll happen is you'll see what foods spike you and which ones don't, and then you just gravitate towards the ones that don't spike you. But if you are going to eat the ones that do spike you, go do a brisk 15-minute walk and
Yeah, yeah.
or make sure you eat your salad or various things before you eat that food, and that we can come back to that too. But there are things you can do to mitigate the spikes.
Yeah.
But back to the spiking itself. Oh, go ahead.
I was going to say, yeah, back to the spiking itself. At a high level, like why do we care about the size of the spike? Why would we maybe recommend to people that you want to avoid things that spike you a lot versus a little? What's the thinking there?
Yeah, in general, there's a correlation between something called time and range. So there's a recommended range to keep your glucose in and your hemoglobin A1C level, which these days is the measure people use for diabetes. So the bottom line is the more you're out of control, if you will, with your glucose, the more you're on your way to becoming diabetic. And it's beyond that too, because you can show these spikes correlate with cardiovascular disease risk. So it's pretty clear. They call these postprandial spikes, post-meal spikes. And so basically, based on that, yeah, you're much more at disease risk. And in general, you want to keep your glucose under control because you're at risk for like everything, cancer, cardiovascular disease especially, but virtually every condition is worse, cancer, et cetera, if you have glucose dysregulation. So we think knowing this early, catching it while you're normal or pre-diabetic, seeing what's going on, excuse me, and then mitigating these spikes should be able to push off, getting diabetes. I'm not going to say it's going to prevent you, but healthier habits should help.
Got it. So in general, when we think about blood glucose, it's like a Goldilocks thing. In general, the body wants it not too high, not too low. And so in general, when we think about the healthy thing, when things are working as they're supposed to, your blood glucose should be as tight as reasonably possible. When it starts going too high too often or too low too often, that can lead to problems, and that does tend to predict various disease states.
Yeah, so for healthy people, we try and keep them between 70 and say 140. As people start losing control, they say, well, try and keep it between 70 and 180 at least. And they call those ranges time and range. You actually touch on another really interesting point, which is not only becoming appreciated now, hypoglycemia. Because when your glucose goes out of control, usually if you you eat something, you're not controlling your glucose well, you tend to make a lot of insulin. And then what'll happen is you'll spike your glucose, but then you'll make a lot of insulin and you overdo it. So then your glucose goes below range.
Yeah.
Which is
and
not
that's when you might feel tired or irritable, brain fog.
People can pass out actually if it gets too low. So you don't want to see. So that's why time and range is a big deal. The bigger problem for most people is the going too high, hyperglycemia. And so that's what everybody, including ourselves, is really trying to try to keep people from, keeping the glucose from going too high. And there are ways of mitigating this. Number one, just eat foods that for you don't spike you. And again, because it's very, very different for different people. The other thing you can do is, as I say, you can actually do a brisk walk, and that will suppress about 15-minute walk. And I wouldn't make it casual. I would walk quickly. That's been shown. And then there are things you can do. And we're still trying to figure this out. We call this eat foods to mitigate those spikes. Okay. And so backing up a little bit, one of the things we showed is that if you're muscle insulin resistant, you'll spike to potatoes and pasta, but not if you're insulin sensitive. So the kind of diabetes you have determines what foods you're going to spike to.
Gotcha. So which tissue is insulin resistant predicts which foods spike your glucose the most.
Muscle's still a biggie. Most people are muscle insulin resistant or beta cell.
Yeah. No, I'm just saying in general that seems to be the case. It's like it's like someone might have muscle insulin resistance and spike to foods A and B. Someone else might be insulin resistant in another organ or tissue system, and they spike to different foods.
Right. So backing up a little bit, Most people will spike the white rice. So watch out that everybody. I just want to throw that out there. Then on the issue of muslins resistance, definitely potatoes is a big one. Pasta. If you're a beta cell defect, you'll also spike to potatoes as well. The bread spikers seem to be associated more. Now, this is getting a little away from it, but people with hypertension,
Hmm.
interestingly enough. So there are these different, and we're still trying to sort all this out. You can tell it's still a work in progress.
Yeah.
But and we're still trying to figure out what other subtypes, like how do you people with incretin defects, what can you do to try to get them to not spike, and which foods are doing that? That we haven't fully sorted out. So it's still in progress.
Yeah.
And then on the mitigation side, we found that protein and fat are particularly good for mitigating spikes on muslins resistant, and same with beta cell defects. Maybe some of the others, surprisingly, fiber, which I thought would be a really good mitigator, it does work. That seems to be very personal. We haven't figured that one out yet.
So, okay, so you gave a bunch of people in this study different foods, and they spiked blood glucose on average to different degrees. And if we read off what you did here, you gave people rice, bread, potatoes, pasta, grapes, beans, and mixed berries. And basically,
Yep.
that's the order for the average glycemic response. So, rice on average spiked people's blood glucose the most, then bread, then potatoes, and so on and so forth. But there's a lot of overlap between these
Oh
foods.
yeah, you could be a bread spiker, for
Yeah.
example.
Yeah.
Yeah, absolutely. That
And
could be your number one problem.
yeah. And a lot of inter-individual variation. And that's what I want to ask you about next is, can you give people a sense for how variable these things that are? Is it that, 80% of people will spike the most to rice, or, give people a sense of the variation here?
Yeah, that's a good question. I'd say it's about 40 to 45% of people would rice would be their top spiking food. Maybe it's about, I'd have to go back and look at the graph, but it's probably about 25% of bread spikers. Potatoes is a big one, I have to say. That does show up probably number two after rice. And I think that's another like 35% or something. But people can be different combinations. I know these numbers won't add up perfectly. Meaning you can be both muslins resistant and a beta cell defect.
Right.
People are more pure one or the other, but most people are combinations of these things.
Yeah. So one person might spike reasonably much to all of these foods, rice, potatoes, pasta, etc. Someone might spike a lot to rice, but not so much to pasta. And you could be sort of any and all of the above.
You got it. Yeah. And bread is really highly variable. Some people will be strong bread spikers, and other people not spike at all, even though they'll spike off the roof to other things. So,
yeah. Were there any features of the patient population that allowed you to clearly segment them based on, are they rice spike or potato spike, the pattern of spiking, the pattern of glycemic response to different foods? Are there any things about did men and women systematically spike differently? Did any other features of the patient population predict the pattern of glycemic response that any individual saw?
Yeah, great question. We didn't see a big difference in sex. We did look for that, but our study's not so huge either. So we may, had I think about 55 people in that first one, although we've added on more now. So there may be a difference, but that has not appeared just yet. There are certain molecular characteristics, certain lipids and triglycerides that will associate with whether you're more likely to be muslins resistant and therefore a potato spiker or not. So we do have some signatures there. And same with, there's a few microbes that are doing this as well. It's an area that we're also diving deeper on, because ultimately at the end of the day, I want to profile a person and say, aha, based on this profile, you're going to be a potato spiker or a bread spiker. We are there, believe it or not, on the curve. So if you wear one of these glucose monitors, and again, you can tell I'm recommending everyone do this because they're really life-changing. Everyone wears them. They just change the way they eat in a good way. And what will happen is these curves, they're very different. So the shape of your curve will be different from my curve, will be different from someone else. And we postulated that was due to the subtype of glucose dysregulation they have. And that turned out to be true because in the end, we could use AI machine learning and make very specific predictions. We're very good for muscle resistant and for beta cell defects. Just from the shape of the curve, we can tell what subtype you have. For the others, we're not there yet. Hepatic insulin resistant and the incretin defect. We're still working on that. We may have to add in other features to build better models. But the cool part about all this is what that means is that you could go to a drugstore, buy one of these glucose monitors, put it on you, drink a glucose shot, and we could tell you what your subtype is. Normally, this would cost thousands of dollars in a research setting. Pretty soon, we hope to have it out there to the real world for free that you could actually just do this yourself, know what subtype you have, and that would put you on the alert that maybe you should watch out for potatoes. Or if you are going to eat those potatoes, again, do that quick walk afterwards or make sure you eat things ahead of time that will suppress that spike.
Yeah. And would the basic idea here, so just to back up for a second, people can buy continuous glucose monitors for like 50, $100, something like that. You can put it on for a week or two, sometimes more. And usually they come with an app these days. So you can put it on and the app will just kind of tell you exactly what the answer is. And it's pretty easy and brainless. It's not too difficult to use. But basically the idea here would be whether or not you're pre-diabetic or diabetic or none of the above, you would probably want to use this to see which foods are driving the biggest glucose spikes in you and basically avoiding those. And certainly if you're pre-diabetic or diabetic, you really want to take great care to avoid the ones causing the biggest spikes.
Yep. Yeah, that's 100% right. Yeah. And then same with the mitigation, you'll want to learn how to mitigate those spikes if you are going to eat these things, or again, and just pick up lifestyle habits that are simple to do. Here I'm 100% conflicted, but I have a company, January AI, that has built a whole program around this that basically, you wear a glucose monitor, they can make all these predictions. They have a 54 million food database, so that this is where the whole field's going. So that you can basically not only take the foods you've eaten and that tell you what to spike, but they'll predict, these other related foods are going to actually cause that same problem. So you should watch out for them as well. So if you, and these days you can go into a restaurant, you can scan the menu and it'll pop up and it'll tell you which foods are going to be the most glycemic for you. And so you actually can again, this is really what precision, in this case, precision glucose control is all about. And I would argue this is where the future should go because we are also different.
Yeah.
We do need to have this information because there's no way you can memorize 54 million foods.
Yeah. One of the things that you mentioned and that you found in this paper is that there were systematic differences between certain types of people. In particular, you saw a difference in the glucose response to rice between Asians and non-Asians. What was the result there, and how do you interpret and think about it?
Yeah, it's a good question. That one still surprises me because, the assumption is that Asians eat more rice and therefore you'd think they'd have better control. Now, I think the rice we eat in today's world is very, very different than it was hundreds of years ago. It's high yield grown, and it only has 8% protein. Whereas ancient rice, people estimate was probably 20% protein.
Oh, wow.
Nutritional content is very, very different today. It's a problem. We grow food for yield, not for nutrition, unfortunately. Right. So basically, I'm guessing that maybe Asians, where it is a more common staple of the diet, were perhaps more attuned to a better, more nutritious rice than today. Otherwise, but like when you ask me what the basis of that is mechanistically, I honestly don't know.
Yeah.
But again, it's someone else had discovered this first. We basically reconfirmed it. And now I would like to try and understand, is it due to the microbiome? Is it due to something, epigenetic about their lifestyle that's different from non-Asians?
Yeah.
I don't know.
And this was a tendency. So whatever the mechanistic basis is, this tendency exists, but it's not absolute. It's not like
Oh,
every
correct.
Asian person spiked the most to white rice and vice versa. And so at the end of the day, this is an individual thing. The only way to know your own response is to measure it with something like a glucose monitor. And you mentioned too, so let's just say someone does that and they realize I'm a rice spiker predominantly, or I spike to bread and potatoes, et cetera, whatever your pattern is. Now you want to avoid those foods or mitigate the response by doing things that cause you to spike less to those foods. Maybe you just love white rice and you don't want to give it up and you're a rice spiker. What can you do to mitigate that big glucose spike? You mentioned walking or moving after a meal here. What about eating foods with other foods, which is something that you looked at in this paper? How does that actually affect the response?
Yeah, so we tested three foods where they ate them 10 minutes before rice in this case, because most people spike the rice. Even if it's not their top spiker, they'll still spike. So we had them eat protein, fat, and fiber before that. And what we discovered was that protein and fat actually did suppress the spike, especially if you're insulin sensitive. It didn't work so well, again, if you had a beta cell defect or a muscle insulin resistance. So the fat kind of lengthen the curve. I don't know if it was basically coating your gut to some extent and therefore slowing down the process. The overall curve came out about the same. But again, that's also highly variable. That depends on the person. So my own guess on the fiber is we have very, very different microbiomes digesting this fiber. And so we handle that very, very differently. In fact, we know that from other fiber studies we've done where we had people eat fiber supplements. And so that would help control that your different microbiome would break down different fibers. Okay, backing up. We all have different microbiomes. Those are the microbes in our gut and our colon. They break down these microbes or the fibers differently. And so the way they handle that will be very, very critical for your glucose spiking, if you will. And one thing that we did notice, I mentioned a couple times that potatoes and pasta, people who are muscle resistant will spike to those. The one thing they have in common is resistant starch. So I think there might be something about the resistant starch. We went on to measure it, and sure enough, it is high in those foods. It's known, but we verified in the foods we had our folks eating. And somehow that in these folks, muscle resistance don't handle that as well. So I don't know if some of the muscle resistant was in part then due to the microbiome and intestinal issues that might actually be all interrelated. And I think this is a general concept backing up. I think we are complex homeostatic systems, humans are, where we're trying to control, our fat and our glucose. They're all interrelated. When you have a lot of glucose, you're trying to store it into your liver. When you need glucose, you break down glycogen to get it back. So we're these homeostatic systems, and we can get off in our metabolism in different ways. And then I think it's important to try and rebalance that. And the way I like the, analogy I like to use, but this may not be a perfect analogy in this case, is a car. They're pretty complicated systems. You want to always be tracking what's going on. So when you see things going off, you can adjust that. And I think that's true for humans. If you see that your metabolism is going off in a certain way, I think we want to try and adjust that to get it back.
Yeah. And there's lots of things in the dashboard of a car that tell you like what's going on, how fast you're going, or if your engine light is on, and those things tell you about something that's going on. But it's also, I think this is also a great place to point out, like the symptom and the cause are often two different things. Like if the check engine light on your car comes out, that doesn't mean you need to change the bulb in the check engine light. It means something else is going on.
Absolutely.
And I guess the bottom line with some of this glycemic response, glucose regulation stuff is it's highly variable between people, and it's highly individual. So at the end of the day, you need to measure it yourself if you want to know what your response is.
Yeah, and we do want to get at mechanisms. And as I say, microbiome accounts for some of this. If that, if you can actually get probiotics, it's a little bit tricky because our microbiome is a community of microbes. So it's hard to just take one probiotic and shift everything. But you could take, especially if you're short on things. So there's a particular microbe called Akkermansia that here I'm not conflict that accompany pendulum cells, and that actually will reduce people's hemoglobin A1C by 1% on average. But for the same thing, for some people, they're amazing responders, others not so much. And I suspect the ones who are good responders have less of that. And this probably helps again rebalance them. But that's one example. When I say things like muscle resistance and these and beta cell defects, we don't understand the underlying cause of that. It's probably a combination of genetics, but my own guess it's a combination of epigenetics. I mentioned how I became diabetic after a viral infection, and I think these viral infections and you may know your microbiome and such gets all set in the first 3 years of life for the most part. You can adjust it, but a lot of it happens in the first 3 years. So you want to be have a good system. Of course, you're getting sick a lot when you're young. And I do think this whole immune microbiome metabolome interplay gets set up pretty early. And I do think you want to, again, keep an eye on that and try and keep it adjusted properly so you can keep your, human body well tuned, so to speak.
Yeah. And I want to shift now. Could be a good transition to the topic of fiber. So fiber ties into the microbiome and variation between people as well. And you mentioned earlier something really important, and this is something that irritates me when a lot of people talk about fibers. They just talk about fiber. You say fiber is good for you, but that's sort of like saying protein is good for you or carbohydrate is good for you. It's like which there's a lot of variation.
Carbohydrate in particular, we know there's really bad ones. Sugar, really good ones. Yes.
So let's start with the basics here. What is fiber, and can you give people a sense for like the different types of fiber and what that even means?
Yeah, sure. So fiber is a polymer of carbohydrate. So you probably know that sugar is made of fructose and in glucose, yes. So anyway, but some fibers like inulin, they're glucose homopolymer. Others like arabinoxylan, they have xylose. So these are different sugars that basically they're put together in a chain. And these chains can be long, they can be short. They can have other molecules in there. There's some very important molecules that over the last few years people have come to appreciate. They're called polyphenols. So phenols, you may know there are certain chemical structures. Polyphenols have multiple of those things. And those turn out to be very, very powerful for your health. It turns out they're antioxidants, probably anti-inflammatories as well. They're cofactors of various things. So anyway, so fibers that have those are probably going to be, well, not probably, they are healthier. And again, I mentioned earlier about charge. Some fibers are more positive, some are negative. And so these fibers, they do multiple things. Number one thing is they feed your gut microbiome. And so we have, as many people here know, we have 37 trillion human cells, but we have over 100 million microbes living in our gut. And so these fibers are feeding those microbes. And these microbes are super important. They make short chain fatty acids. They make essential vitamins like B12, all these things. So this is why people say fiber is good for you. But different fibers are going to feed different microbes and behave very, very differently. And so one study we ran is we had people eat a supplement of this arabinoxylan, this thing that's found in Physium husk, or inulin, which is chicory pea root. Again, these are very, very common fibers you'll see out there, just to see what their effect would be. And sure enough, you do see hundreds of molecules changing with each of these when they just take it as a supplement. And one of the things that we did pin down was that ribosylane, but not inulin, does lower cholesterol quite dramatically, actually. Neither had an effect on glucose in general. And so that wasn't so clear from the literature when we went into this. Even though in general for ribosylane, people went down in their cholesterol, there were some people who didn't respond at all, meaning they had no effect. And others, and those same people, they would go down with inulin, by the way. So we're assuming now there we don't have it all worked out that that's probably due. We're running a subsequent study to actually see. We're assuming it's due to the microbiome and how well they can handle those fibers. So we all have different microbiomes, so we're probably handling these fibers very, very differently. And so a fiber that's good for you may not be good for me. The party line is that a mixture of fibers is better, and I think that's probably generally true, but I think there are many exceptions to that too, from what we're seeing.
Right, right. So the devil's in the details. Again,
Yeah,
individual responses are very different, and you can't really make an umbrella statement, certainly about all fiber for all people.
correct. Yeah.
Yeah, yeah.
Well, again, what's good for you may not be good for me.
Right. But just to tie this together, so when we use the term carbohydrate, by default, we basically mean carbohydrates that we and our cells can chew up and digest. Fiber is carbohydrate, but it's basically a form of carbohydrate that our cells can't really break down. And so the microbes break them down instead.
Correct. Yeah. Yep. Yes.
Okay.
Exactly.
This also brings us to another interesting topic, which you have studied, which is short chain fatty acids. I feel like this is probably something where people maybe haven't heard this term as much. It's a very important area in research. I know that a lot of people are working on this now. Let's start off with what are short chain fatty acids and what's their relationship to fiber in general?
Yeah, they're these small molecules that, again, get made by your gut. Butyrate, propionate, things like this. Um, yeah, and what will happen is that when you feed your microbiome with fiber, certain microbes are very, very good at making these short chain fatty acids. And another group, Yiming Shao at University of Chicago, had shown that these short chain fatty acids can wind up on histones, which are key molecules that regulate gene expression. And it kind of sat there. And by the way, backing up, short chain fatty acids, again, very, very clear. They're good for immune health. They have all kinds of, beneficial effects. But exactly how they exert those is really not clear.
Yeah.
You need them for your gut integrity, all this sort of stuff.
Yeah. But basically, short chain fatty acid, it's a small fatty acid, and we're predominantly getting them from microbial metabolism in our gut.
Correct. And very specific microbes, not just one, several types of microbes are a really great producer of these. So when you feed those microbes with your microbiome, with your fiber, they'll start producing these short chain fatty acids, and that's good for you. So what wasn't so clear is the mechanism by which these short chain fatty acids work. And so a lot of people thought, well, they work because they're inhibiting another kind of modification that shows up on these histones. It's called acetylation. And most people assume they were affecting the enzymes that affect acetylation, and which it probably still has some effect that way. But we sort of recognize, based on the work of others, that maybe what they're really doing is directly affecting gene expression. And then the question is, what genes are getting affected? How is this useful to you? And so what we discovered is that these short chain fatty acids, we took intestinal cells and liver and other cells, actually, but mostly intestinal cells relevant for this discussion, and basically treated them with short chain fatty acids, propionate and butyrate, which again would get made by fiber. And we saw that they wound up on histones at very specific genes. And those genes are involved in gut integrity and transporters and things that should really help make a good, healthy gut.
Got it. So short chain fatty acids are produced by microbes in the gut once they get their hands on some of the fiber we might be eating, if those microbes are present in our gut. And then these short chain fatty acids are somehow getting into some of our cells, getting into the nucleus and interacting directly with the chromosomes and the genes in our cells.
And turning on the genes you want turned on. Exactly. So we think we've gotten at a molecular explanation for how fiber works, basically. And I should say we just showed intestinal cells, but it's pretty clear these short chain fatty acids spread throughout your body. They wind up in your bloodstream. So they're probably promoting health all the way around, not just your intestinal cells. We pinned it down in the intestinal cells. And then we saw a lot of this was dysregulated in cancer. So we looked at cancer intestinal cells. And sure enough, you're starting to see things affecting cell proliferation and such being affected by short chain fatty acids. And so I think, again, while you're healthy, this is super, super valuable for keeping a healthy gut. But when things go off, they may not be helpful. They may actually be, in fact, accelerating problems. And I should back up a little bit. Why do you want a healthy gut? Well, if you have a leaky gut, you may know microbes and things getting into your bloodstream. That's bad news. That's not good. You start making immune reaction. It's associated with all kinds of
Yeah. You want your gut microbes in the gut. You don't want them to get through the gut. And there's a barrier there. There's mucus and different structures that
very tight barrier. Yeah.
So all of that is most of that is related ultimately to things like gene expression. So these intestinal cells have to have the right genes on and the right pattern and the right amount in order to maintain that barrier and not have a leaky gut, for example.
You got it. Yep.
So we think the production of short chain fatty acids by the microbes is helping regulate the pattern of gene expression in intestinal cells, at least. And part of what that does that's beneficial is it maintains the integrity of your gut, including the barrier between your gut, the inside, and the microbes living in the gut lumen.
Yeah, basically a nice tight gut, if you will. Yep. And again, that is just important for so many different things, for preventing autoimmune disease and all kinds of important situations there.
Are short chain fatty acids only produced from microbial metabolism, or can we eat them directly? Are there other possible sources of these things?
Yeah, you can eat them directly. And we're running studies now where we're feeding these to mice to see what will directly happen. And the literature is a little bit conflicting out there. I'm going to guess it's coming down to dosing, getting this dose just right. Because sometimes if you have too much of one thing, that's bad news. As you may know, vitamin D3 now, it's very, very clear taking vitamin D3 as a supplement is good. That has good health effects. But there's some studies coming out now that too much, way too much, by the way, not just a little too much, is not good for you. And my guess is that that's going to be true for the short chain fatty acids. Get them just right, so that you have optimization throughout your body. But there are studies out there that show positive effects of feeding people butyrate. And I might also point out that our data also showed slight differences between the short chain fatty acids as well. So I want to study that further, but propionate was different from butyrate, which is different from acetate, if you will.
Yeah.
So 3 different forms there.
Yeah. So butyrate is one of the main short chain fatty acids produced from microbial metabolism of fiber in our gut. That sounds pretty close to beta hydroxybutyrate, which is the thing that goes up when you go into ketosis, or which some people supplement with as well. Is there a big difference between BHB, beta hydroxybutyrate, and butyrate? Is it possible that people in a ketogenic state or in ketosis are getting some of that short chain fatty acid benefit from BHB rather than butyrate?
Yeah, it's a great question. They're not identical. They're the same. And this is an area that goes outside my expertise, so I don't know the answer for sure. But what you say is very true that, a lot of mental health conditions, bipolar specifically, is controlled, seems to be well managed by a ketogenic diet. And certainly, the hydroxybutyrate and things like that seem to be are likely to be a key component of that. But I predict it's going to be a mixture of things, and the exact relationship, as you point out, between hydroxybutyrate and butyrate, they can interconvert depending, what's doing it. But how well that's all worked out, it may be better worked out than I know. I just don't know. But these are things that should certainly get studied, because at the end we want to have precision diets for people.
Yeah. One of the things you looked at in this paper as well was short chain fatty acids in colorectal cancer, colon cancer. I've done a few episodes on this, but one of them was really interesting, looked at lipid dysregulation in human tissues of colon cancer. And there's some interesting stuff there. You guys were looking at colon cancer in animals, I believe, in the context of these short chain fatty acids coming from fiber metabolized by microbes. One of the reasons this topic is sort of relevant or top of mind for some people is, I believe colon cancer today is one of the cancers that's actually rising the fastest in young people. Can you give us some of the context there for why that might be happening and why people are studying that?
My guess is it's diet. I really think the Western diet is so unhealthy, and we start at a very young age giving people the wrong foods. So I really think, I mean, obesity is a good example, right? With huge numbers of people being obese right now, over 50% of the U.S. population is overweight, dramatically overweight or obese, which is not good. And those numbers are rising, by the way. And the same numbers for diabetes. They parallel pretty well. So meaning 38% of the US population is pre-diabetic, 11.6% is diabetic. So again, about half the population pre-diabetic or diabetic. So we've really got to get this under control. And I think it's a bad diet, but that's my guess starting early on. It's very, very clear. Believe it or not, we have some other studies where we study colon cancer, early stage colon cancer, in an interesting model system. It's called familial adenomatous polyposis. I know that's a mouthful, FAP for short. But individuals with that have a mutation that puts them at high risk, and they wind up having all these polyps that are thought to represent early stage cancer. So we've been studying those, and what you discover right away, even in the smallest little polyps, which are these precancers, there's a lot of inflammation going on. So I think inflammation is very, very tightly associated with cancer. That's been shown in other systems. And so, again, feeding your immune system to keep it, and your microbes to keep things anti-inflammatory, we think is a big deal. So I do think we've got to get people's diet better under control, less inflammatory. Sugar winds up being hyper-inflammatory for your immune system. Yeah, you'll see there's a protein called C-reactive protein. People who are diabetic have high levels of this, and other things as well, cytokines. I mean, it's very, very clear. So I do believe we have to get proper diets started early in kids, and get rid of the sugar that's just too prevalent in the U.S. population.
Yeah. Can you dig into the colon cancer short chain fatty acid result a little bit more? Were you depriving certain animals of short chain fatty acids by depriving them of fiber, and this was inducing colon cancer, or how did that part of the experiment work?
Yeah, that one we did by changing their diet. We put them on a high fat diet or a high carb diet. High carb being, again, not a good diet. And that actually induced much more inflammation. We have other studies, not fully published, but basically then the genes that got affected were more, if you will, by the short chain fatty acids, more of these things that promote cancer, MYC and stuff.
So the pattern of gene expression change that the short chain fatty acids induced depended on the greater dietary context that was given to the animals.
Correct. Yeah.
Got it. So it's not just short chain fatty acids make good gene expression changes happen, period. It's the other nutrients and things in the diet in general affect how the short chain fatty acids turn on and turn off different genes.
It is. Yeah. I mean, in that case, they were extreme diets, but it definitely had different effects. Mm-hmm. Yeah.
And when you say extreme diet, high carb diet, what exactly does that mean? How high was high, and what type of carbohydrates?
Oh, I'd have to go back and look at the numbers, but they're primarily carbs, and they were things that the mice would digest, not exactly healthy carbs, as I recall. We didn't give them straight up sugar, but we gave them things that basically converted to sugars very easily. And the high fat diet was much more, again, mostly very, very low carb, but generally very, very fatty diet, like you would do if you were doing a ketogenic diet.
Got it. So it's very fatty. So one thing to talk about there that I think could be relevant is, other people have done work. Tim Yateman and Ganesh Halliday, I've had them on the podcast. I highly encourage people to look at that if they're interested in diet and colon cancer. They found that in human colon cancer tissue, those tissues were basically characterized by a state of lipid dysregulation. There was a lot of inflammatory lipid stuff in the colon cancer tissue. And mostly those inflammatory lipids were those that are derived from omega-6 polyunsaturated fatty acids, what we might call seed oil fats. And they really thought that may have been the dietary explanation there, or could be one factor. As you may know, Michael, when we talk about a high-fat diet in humans or the standard American diet, one of the key features of it is high levels of omega-6 polyunsaturated fatty acids. What can you tell us there about the dietary fats in this high fat diet that you and others have used that might tie into these things.
Yeah, no, good point. So we can break this down further as you're doing. So omega-3s, let's back up a little bit. They're very prevalent in your brain and your eyes, actually, these omega-3s. And everyone agrees they're very, very healthy.
Omega-3, that would be like fish oil fat, basically.
I was just about to say that. So fish oil, some of these other related things. And so I'm a big fan of those. In fact, I take two, 1 gram tablets or maybe it's more than that. Anyway, the big tablets every day. And I've been doing it for a while. And
I think that's important to highlight, though. You supplement with it, but we're talking about a couple of grams per day. So,
oh yeah, this is a big. These are a fair amount, and there's plenty of studies out there. If we back up a little bit, and this is kind of a big deal to me. There's a lot of studies associating, various nutrients like omega-3s with cognition and health. And those studies are very, very clear. And these days, there's big ones that come out between Mediterranean and vegan diets being good for health and avoiding ultra-processed foods, not just for, carbs like the cakes and things, but also meats, where we're going with this conversation. Avoid those ultra-processed meats, which have a lot of unhealthy fats in them. So basically, omega-3s, again, good. Omega-6s are now becoming a little more controversial. There's no question. There's plenty of correlations between omega-3s and unhealthy things that can happen. Like
between omega-6s and unhealthy things.
Sorry, omega-6s. Thanks for catching that. Yeah. So, for example, in your gut, causing inflammation and various things. There may be contexts in which omega-6s are actually beneficial. So I think that's pretty recent. So I think this still needs to be sorted out a little better. But on the whole, a lot of the omega-6s are not so good. And so I think and they can be associated with inflammation and things. Now, one direction I was going to go with this is that a lot of people take these as supplements, which I do.
Omega-3s.
Omega-3s, yes, and also lots of other vitamins. I have a whole list. In fact, I'll put it up on my website. I've been meaning to do that, but I take, antioxidants, coenzyme Q, the sort of thing. And many of these have not been directly shown as supplements to improve health.
Right.
They're kind of like, and this is where the controversy exists, that people say, and it's true, you probably pee out most of these supplements when you take them at these incredible doses. But clearly some level of this stuff gets in. And for some people, it's a pretty big deal. Like, a simple one like magnesium that I take at night, that actually is a muscle relaxant and it helps a lot. It's good for your sleep. But for some people, it's almost essential for their sleep. Like they're, for whatever reason, deficient. And then they take these tablets and it really helps them. So supplements is a very tricky area. It comes back to this, personalized response again. And also the fact that there aren't necessarily amazing studies out there, large studies that show they work. There are now for vitamin D3. And so I think this is where some of the controversy exists, that people say, well, this is a supplement, hasn't shown to work. But I still take a lot of them because there's still, I think, a lot of common sense around this. Like I say, taking antioxidants, anti-inflammatories is a good thing.
Yeah.
Obviously, it's good if you can get it straight up from your food. It's a great source. But for many of us, I take nitric oxide beetroot extract, which is known to promote endothelial function.
Yeah. Well, I think that this also just fits in with the greater theme of, when you're thinking about any given nutrient or compound or antioxidant or whatever that's being ingested, the greater dietary context pretty much always matters. So, we talked about short chain fatty acids, and the effect they have on gene expression depends on the macronutrient profile that they're embedded in the diet. Whether or not you take, a gram of a supplement of fish oil might not get absorbed or taken in by your body the same way as if you eat a gram from a salmon fillet or something like this. So you always have to think about the greater context these things are embedded in.
Yeah, and there's a few studies that look at this, like vitamin C, when gotten from a lemon, apparently there's a binding protein that helps its uptake and things like this. So 100% agree. But if the issues of not getting something versus always putting it in the right context, well, there can still be value by supplements, at least in some cases. The bottom line is we just need a lot more studies out there to really pin down what does work and what doesn't work. Then, of course, there's interactions between supplements. And this is where I think AI can really kick in. I know AI is very hyped. I'm a big believer in it. But we had some people doing very simple thing. We actually teach a friend of mine, Rajan Neg, and I teach a vibe coding and longevity class. So you don't have to know anything. You just walk up, come up with an idea, and as you probably know, vibe coding is where things will code for you, and in 15 minutes you can come up with your own algorithm. So he came up with this idea. Why don't we see what the interaction between supplements is? And so he vibe coded, if you will. I mean, he told one of these, I think he used Claude AI, I forgot which one, but he used one of these things to basically come up with an app that basically, and then he loaded his apps in and said, I take zinc and I take, what was it, zinc and magnesium. Yeah. Is there an interaction? He loaded them all in there, and it basically spit out and said, well, you should take those at least 6 hours apart because they use the same transporter.
Right.
There's no way any normal person is going to know this, but somehow AI obviously found the paper, and you could go back and find the paper and realize they use the same transporter. Well, it's logical then to take them 6 hours apart, right? And so this is where I see this whole field going, that we can use this information. We still need a lot more good information out there. Don't get me wrong, of the sorts that I've been describing earlier, the studies around fiber, supplements, things like this. But then I think we can use AI to better manage people's health and just do things right. And one of my big bugaboos these days is if you have cancer, what should you eat afterwards? Nobody will guide you, right?
Yeah.
Good recommendations. Or if you have a heart attack, what should your exercise regime be after this? And I guess I can fully disclose, I just had a hernia operation 5 weeks ago. So you're not supposed to, don't lift anything over 10 pounds. Now I'm trying to figure out what is the right exercise. I used to lift weights every day.
Right.
To build, because I'm on GLPs, which do cause muscle mass loss. So I'm trying to actually, and so of course I miss losing that, and of course I'm losing muscle mass.
Right. So the question is like, how often can I lift? How heavy should it be, and so forth.
I want to know exactly what I can do to get back there without damaging the chances of my, my hernia operation. I guess this is full disclosure here on the show, but anyway, you get the idea. We want to really know what to do, and this stuff is all figure outable, if that's a word. We should be able to drill in, just like back at the beginning of our conversation, exactly what fiber is for what person. And it will be personal, but there should be ways to measure this, have, generalized recommendations, but then very specific things that you watch out for to best personalize it for yourself.
Yeah. And, coming back to the glycemic response and continuous glucose monitors, one thing I love about that approach is it's accessible to anyone. It's relatively affordable. Anyone can buy this online. And it's sort of like a beacon or like an all-in measure. You don't need to know 1,000 different nutrients and how they all interact and get like lost in the weeds. Nobody is going to want a really high glycemic response. No one's going to want their glucose response to spike too much or too often or whatever. And so it's like one good marker that is easy for even the average person to look at and give them something to aim for. Let's say that,
and it's very visual in real time, if I
can
interject. And I think that's why it's so effective. You see it right while it's happening.
Right.
You eat that McDonald's shake and watch glucose go up over 400.
Yeah. What's tough about blood work, I think, for a lot of people is I can get my blood work done today and get a whole bunch of numbers, but is something up or down because of what I ate yesterday, or is it the general? Like, it's hard to pinpoint it to anything. Right. Whereas you're saying with a CGM, a glucose monitor, after you eat the rice or the potatoes or whatever it is, you can see how much it's going to go up. Let's say someone wanted to kind of almost recapitulate what you did in that one study and figure out, like, am I more of a rice and potato spiker, am I bread spiker, grapes, whatever it is. What is a simple way that an ordinary person might go about that? For example, would a good strategy be to eat a simple breakfast day by day so that, like, you're going to bed, you're sleeping overnight, your stomach's relatively empty, and then I'm going to have rice or breakfast, look at my CGM. Maybe the next day I do it with potatoes. How would you think about going about that for an ordinary person if they wanted to be somewhat systematic about it?
Yeah, the chemist in me says to do exactly what you just said. Break it down one at a time to know exactly what those foods are doing. Now, I know there's a lot of variables there, but nonetheless, you can get a pretty good idea which going to work. By the way, we're about to run a study with different types of potatoes, French fries versus mashed potatoes versus baked potatoes, this kind of thing. So that's going to be a lot of fun. But I think it's to do it exactly the way you say. Again, start with a clean slate and then see how to mitigate that. What personal foods, the fat, the protein, the salad would mitigate those spikes. So I think that's a good way to go. One thing that we're going to try and have people do, again, we're just repeating the study, although I'm pretty confident because we did it the way we did it with several different ways. But we'll have people drink a glucose shot, and then they can see from the shape of the curve what we expect.
Right, right.
We can then know their subtype, and that will make recommendations, although they should still do it for themselves, just the way you're saying, because maybe there's other mitigating factors we haven't identified yet, like that doesn't, we'll predict whether you're a potato spiker, but then you could actually see it yourself. And again, it's very visual, so that would be nice to know. Yeah. And by the way, I do predict there are circadian effects. We have not looked at those much. And so even if you had fasted and ate that potato at dinner, you may or may not be the same as that one in the morning.
Right, right, right. Yeah. So all things to consider. What's nice about the CGM again is you can wear it for a couple weeks, and so you can do both. You can look after dinner, you can look in the morning, and everything. I've done it before, just so people know. It's kind of fun. I mean, it's kind of
really
cool
fun.
to see what happens. Yeah.
Yeah. No,
Um,
it's highly recommend. It's just plain old good for your health.
yeah. So we've talked a lot about carbohydrates and the glycemic response. We've talked about short chain fatty acids. I want to talk a little bit more about fats and lipidomics. You've done some interesting work in various ways in this realm. Let's just start basics here. What is lipidomics, and what does this field do, and how does it work?
Yeah, it's one of the less studied areas, to be honest. And just like we were talking about a little bit about the controversy on omega-6s, those can be, well, fatty acids, but they can be lipids for sure. It's a very, very heterogeneous class of molecules. You have sphingolipids, ceramides. These are all these different compounds, if you will, all phospholipids of phosphatidylcholine, phosphatidylethanolamine.
Yeah, all kinds of lipids, lots and lots of different ones.
It's literally thousands, and the complete map is not clear. And I won't call it infinite, but it's nearly infinite when you think about it because of the way they're combined and such on like triglycerides means there's three chains of these things coming off. So you can have, and the chains can be of different length and so on and so forth. So there's short chain fatty acids, long chain, and they're just important in so many different ways. So some of them are energy molecules. For example, when you exercise, you use some of your short chain fatty acids, actually, we think middle chain fatty acids as an energy source. And there's something called acylcarnitines that are also used as well. So you have all these. Yeah, so some of them are energy source. Many of them are signaling molecules, and this is not at all fully worked out, meaning they'll have global signaling effects. So think of cytokines, lipids as a group of molecules that may have just as pronounced effect as cytokines that's just unexplored.
Yeah, and when we say signaling molecules, we basically mean they're not just used for structure, like in a cell membrane. They're not just used to burn for energy, which is one way that can be used, but they can plug into different receptors and information pathways in cells to affect the behavior of the cell and which genes are being expressed and that type of thing.
Yeah, we have a little known study out there where we had done a pull down of proteins to see what fraction would have a small molecule along with them. It was like 25%, some ridiculously high number. And a lot of those were actually lipids and short chain fatty acids and things. So we think that there's just a lot of this interaction. Again, all these molecules in the cell, they certainly have things they're famous for, ATP and stuff like that. But then they probably have lots of other roles that aren't so clear. And as we learn more and more, and lipids have generally been, I mean, I think people used to just throw their arms up in the air because they're so complicated. There's so many different forms. But now people kind of look at them as a class because at some level you can learn something as a class of molecules like the triglycerides or things like that. And we're trying to drive into the deeper detail, like the ceramides are clearly showing up as signaling molecules. Sphingolipids, more and more people are appreciating as having important effects, and some of them actually probably bind your chromatin and have direct effects on gene expression. I think that's pretty clear. So yeah, there's just going to be lots of functions out there. So we've done one of the studies we're running. We've been profiling a group of people now for about 13 and a half years, almost 14 now. I'm one of them. We've been doing me for over 16 years of profiling them very, very deeply for their metabolites, their lipids and things. And then we try and correlate these things with lifestyle. So the goal really is to make some of these associations. They start out as associations, but I think we can get more into what's called causal relationships, not just understanding that this is associated with that, but this may cause that. And there's something called mediation analysis or causal inference, where you can basically, say, well, this microbe is associated with this lipid that's associated with this behavior or phenotype. And so I think groups like ours are trying to tease these out. We do see a lot of these kinds of things. We don't really always know what they're kind of chemical formulas. We don't always know what those lipids or metabolites are. So I think it's a very, very understudied area.
You've done some work. We were chatting a little bit ahead of time in the realm of maternal health and pregnancy. And this is a fascinating and obviously very important area, probably an understudied area as well. And there's lots of stuff to dig into here. Lots of fascinating work going on in this realm. But what are you doing today, or what have you done recently in regards to maternal health and pregnancy, and sort of how a mother's diet might impact her developing offspring?
Yeah, so we've done a lot. Initially, we did a lot of metabolomics profiling, and there are a lot of lipids in there too. And it's very, very clear there's a lot of hormones that change dramatically throughout pregnancy, for example. And they generally go back to normal in the mom after they deliver. Not 100%, but most of them do. I think one conclusion that comes out of that is there's a lot of hormones that people probably don't know about that they're under study. We all think about the estrogens, or several of those, and some of the progesterones and stuff like that that are actually also women. But there's a lot of other things that are just embedded in this profile that I suspect are going to have useful behavioral functions and activities. And then we have looked more recently. This one's not published, trying to look at the cord blood. Which is kind of cool because that's the interchange, for those who don't know, between the mom and the fetus. And then at birth, the cord blood's normally discarded, although these days people often save the stem cells from that. I did for my kids, actually, for what that's worth. And by actually looking at the arterial versus the venous flow in that, you can actually see what the fetus is consuming. It's kind of interesting. You can see very specific molecules, including specific lipids, a lot of the
and is this
fatty acids.
Is this done in humans?
Yeah, so it's in humans. So you can take the leftover cord from after birth and pull out get the plasma from that cord blood. And if you follow arterial versus venous, you can see again by one's going into the fetus and one's coming out. And the delta is thought to then be what the fetus is consuming. It's kind of fascinating. And what we're seeing is what metabolites the fetus uses. And there are interesting metabolites, a lot you'd expect, but then there's a number of lipids that are showing up. So back to your lipid part. And then we also see a lot of things exchanging between the mom and the fetus, like what kinds of drugs a mom might take, which ones wind up in the fetus and which ones don't. Now, we don't always know their health consequences of this, but at least we'll see, well, if the mom does take this, this might be what, this is sharp to the fetus. It could have an effect. And actually, we have a separate area where somebody's studying the effects of epilepsy drugs that moms take, and what does that do to the fetus? Most of this area is pretty understudied. I guess going back to the original thing, though, there's just thousands of changes that occur throughout pregnancy, and the whole metabolic profile shifts in moms, and that's not surprising. There's just a lot going on.
And if we started to learn anything that gives us, directional knowledge about things that are driving, good or bad outcomes for the developing child. So for example, are there certain things that jump out of a metabolomics analysis that correlate with preterm birth or other complications and things like that?
Yeah, they were finding quite a few molecules, both metabolites and lipids, that actually are associated with preterm birth. So I'm hoping we could set that up as a basically as an assay to flag preterm birth. We've done some and found specific molecules associated with preeclampsia as well. And so the goal there would be to do just that, to have this. Interestingly, in the cord blood, I know I'm bouncing around a little bit, but in the cord blood experiment, we actually have a signature for a healthy baby, meaning that in general, these kids don't have complications. I think it's out to 6 months and a year after birth. So if you think about it, we tend to always look for complications, but it's kind of nice to have a signature. Well, at least from this test, we don't see signs of a health problem. We're expecting this to be a healthy baby for the next bit. It doesn't mean it might help keep a few moms feeling a little appeased there that things look good. So it'd be nice to have more healthy assays and not just sick assays, right?
Yeah.
And so we're very excited about that. And going back to the profiling through pregnancy, again, the number one thing that really struck me is just it's thousands of changes occur throughout pregnancy. The ones that caught our eye a lot are these different hormones. So it's pretty fascinating.
Yeah. What are some of the things that are on the horizon for your lab, things that you're going to probably publish on in the near future that you're particularly excited about?
Well, the cord blood one I mentioned for sure. We want to dissect this personalized nutrition a lot more. I guess we want to understand the cause of these glucose dysregulation subphenotypes. It's nice to say muscle insulin resistant. My own guess is some of this is epigenetic changes, that there are shifts in people's modification of their metabolic genes. That happened to me. I didn't get into this, but after I became diabetic, the promoters of about 100 or more metabolic genes got methylated, got modified, and we think that's affecting gene expression. We see shifts. They're subtle, but when you do it over 100 genes, and the odds are that's what's causing my diabetes. And then I'm guessing I'd love to see more about how epigenetic perturbations, say viral infections, other things, do shift people's metabolic control. So there is that one. We're doing personalized nutrition control, like we talked about. I want to understand better what are the subphenotypes. We mentioned the different potatoes affecting people, but how do you mitigate those? And we just tested 3 things initially. I want to test more, and I want to get into combinatorics. We're also getting, in this, we just started publishing on a little bit, but mental health. We don't really profile. Mental health is very tied to nutrition. Again, we talked about bipolar and the way you control that being ketogenic diets, and that really seems to work well. But I don't think, and I'm a big believer in the gut-brain axis. So I think we want to understand better. We don't have good markers for mental health for depression, anxiety. It's always surveys, right? And surveys, it's not a great way to measure things. How do you know when you're treating somebody if they're getting better? So we need good markers. And we didn't talk about this other aspect. We do a lot with the wearables, the smartwatches, things, and this sort of thing. So I think there's a lot of digital markers that can be brought to the mental health space, and that can be very, very valuable for tracking people's mental health, so that you can actually have alarm. If you are starting to get anxious, maybe wait. Go calm down a little bit, or maybe it's time for a vacation or something. Things that you might do to help improve people's mental health. Often it's probably just sleep more. but I think we can probably do a lot. We can understand better mental health better, I think, by measuring it better, and we can also, again, I hope, intervene and keep people healthier and happier if we can measure these things better.
Is there anything that you want to reiterate from our discussion, or any final thoughts you want to leave people with about anything we talked about this general subject of personalized nutrition and medicine and individual variability and these types of things?
No, I think it's really important to know what's going on. If people always ask me if there's one thing you can measure, I'd say go get a CGM because I think they are so valuable. And people always wait till it's way too late. And then it's just hard. This is true for any disease. It's hard to cure disease. It's a lot easier to try and keep people to stay healthy. So I'm a big believer that you should get one of these things. I'm a big believer. I wish I knew 50 years ago what I know now. I would probably have had very different behavioral patterns than now I think I'm generally pretty good. I can do better on my sleep, but they're not too bad. But I wish I'd done this earlier. Perhaps I never would have gotten diabetic. Who knows?
All right. Well, Professor Michael Snyder, thank you very much for your time. I really appreciate it. You do a lot of stuff, obviously. We didn't touch on nearly everything that you work on. So I encourage people to check out your work, and I look forward to following you along.
Thanks. Fun to be here.
Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio, formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.